All right, so sorry, just making sure this works. All right, so today we're going to look a little bit more closely at exactly how substances get across the plasma membrane. We talked about already how the plasma membrane can only naturally let things that are small or nonpolar through. And we talked about how proteins, integral channel proteins, are necessary to let larger or polar molecules through. But we're going to talk a little bit more about exactly how that happens today, looking at osmosis and diffusion and things like that. So hopefully you might remember some of this from freshman biology, but otherwise we'll go over kind of the nuts and bolts today. So the main way that things pass from one side of the plasma membrane to the other is through diffusion. And so this is like how small nonpolar substances go right through the plasma membrane. Right? It has to do with the second law of thermodynamics, okay? And that says that the universe tends towards entropy, increased entropy or disorder. So what that means is that if I start out with a bunch of molecules of dye on the inside of the plasma membrane, and they are able to freely cross the plasma membrane. Let's say that they're small nonpolar molecules. That because of the collisions that they have randomly moving around, they will tend to space out and eventually become equally concentrated on both sides. And this idea is called diffusion, or the idea that because of these random collisions, substances will tend to move or spread out from areas of high concentration to areas where they're less concentrated. So in this sequence of pictures, at the beginning, or at the picture farthest to the left, the dye molecules were highly concentrated on the left, and because there was an increased amount of random collisions between the dye molecules, they started to space out and move to the right side where they were less concentrated until eventually they reached an equilibrium point where just as many molecules of dye were involved in collisions that pushed them to this side as were molecules of dye in being collided with that pushed them to the other side. So the back and forth balanced out. So simple diffusion like that moves molecules from high to low concentrations. And it's an example of what we call passive transport because no energy is needed. We say that it happens spontaneously, all on its own. So as an example, like we saw in this animation, all of these purple molecules were concentrated on this side of the screen, and over time they spread out, diffusion. Or, I mean, this also happens with water. And when diffusion happens, and the substance that's being diffused is water, we call that, give it a special name, we say osmosis. So osmosis is just a special case of diffusion when the substance that is diffusing is water. For substances that are bigger or polar, that can't just diffuse through the plasma membrane on their own, there is facilitated diffusion, which just means that there is one of those channel proteins that we talked about that will help the that occur. So it's still moving from high to low, except in this case, it's moving through this doorway, this channel that's created by the protein. It is also an example of passive transport because no energy is needed for it to move from one side to the other. It just needs this little hole doorway in the channel protein. So still going from high concentration to low. On the other side, cells will often need to move substances against their concentration gradient. So in the opposite direction, move from an area where their concentration is low to an area where the concentration is high. And this is called active transport. Okay? It requires energy. So moving something from low to high requires the expenditure of energy because it's opposite the direction it'll naturally want to go through diffusion. And usually special transport proteins that pump or push those substances from low to high are needed, protein pumps, to accomplish active transport. As we'll talk about later, this process requires energy in the form of ATP, adenosine triphosphate, which is kind of the energy currency of the cell that you might have heard about before. All right, so there are many different models of active transport, and some of them you can see below. In each case, there's like a protein. We'll go back and we'll see that animation again. In each case, there's a protein that kind of changes shape uh, to allow substances to go in and out, to pump substances from low concentration to high concentration. So remembering that active transport requires these proteins to pump substances from low to high, and then it requires ATP energy. So as a review, passive transport includes simple diffusion, diffusion of nonpolar hydrophobic substances like lipids from high to low along the concentration gradient, or facilitated transport, where the diffusion of polar hydrophilic molecules occur through protein channels, again, from high to low concentrations. And then there is active transport, which goes against the concentration gradient, the way things would naturally want to diffuse from low to high. And they use protein pumps that require ATP energy to work. All right, so that's the basics of membrane transport. This is kind of a picture summary of simple diffusion, facilitated diffusion, and active transport right here. How about really large molecules? Moving really large substances in and out of the cell requires endocytosis and exocytosis through vesicles and vacuoles. In endocytosis, what in endocytosis and exocytosis, basically these big vesicles of substances, either inside or outside of the cell, merge with the plasma membrane and spill their contents either into or out of the cell, moving a bulk amount of substance into or out of the cell at one time. And so that's why this is called bulk transport. You might kind of think of phagocytosis like the endocytosis that occurs in white blood cells when they surround and eat a pathogen. So here's endocytosis, so something we want to move into the cell, something that we want to move into the cell is being swallowed up by a vesicle of the cell and eventually goes inside. So here's an example of endocytosis. And then exocytosis would be just if we went the opposite direction, starting with it already in the vesicle and then merging with the plasma membrane and spilling its contents out of the cell, So, the last thing I want to talk about was the special case of water, like we said, that moves through diffusion across the plasma membrane. So osmosis is just the diffusion of water, like we said. Water is very important to life, so we kind of talk about water separately. Diffusion of water still occurs from high concentration of water to low concentration of water. So osmosis, same with all the other types of diffusion, high concentration of water to low concentration of water. So as an example here, Both of these sides of this tube are at the same level, and there is a permeable membrane here that's only permeable to water. So these purple molecules can't go from one side to the other. Now, if we think about the concentration of water, these purple molecules take up space, which means there's less space, there's less water on the right side since more of it is purple molecules, and more water on the left side. So higher concentration over here of water, lower concentration of over here of water. And so water will move via osmosis from the left side of this tube to the right side of this tube, creating this unequal water level. And we can actually do an experiment exactly like that that will have that same result. So the concentration of water is all important. The direction of osmosis is determined by comparing the total solute, what's dissolved in the water concentration. A hypertonic solution has more solute in it and less water. So for example, this side compared to this one, right? These terms are always relative to one another. A hypertonic solution has more solute dissolved in it and less water than what we're comparing it to. And a hypotonic solution will have more water and less solute compared to whatever comparing it to. So water will always move from the hypotonic solution into the hypertonic solution if they're separated by a membrane that's only permeable to water. If we would have two solutions that have an equal concentration of solute and equal concentration of water, we would call those two solutions isotonic. But the important thing to remember is these three terms are not absolute. For example, if I made another one over here, this solution could be hypertonic to this one, but hypotonic to the next one if I drew another one over here that had even more solute in it. So they're completely relative terms. Cell survival depends on balancing water uptake and loss. So for example, if we put a cell into a hypotonic solution and just let it sit there, it would take in so much water that it could burst. Or if we put it in a hypertonic solution, it could lose so much water that it could shrivel up. All right, and that would not be good for the cell. So cells work really hard with, for example, uh, transported proteins to maintain an isotonic environment in which their inside and outside concentrations of water and dissolved solutes are about the same. All right, so that's it for today. We wanted to learn about transport and then specifically look at osmosis a little bit more closely.